Hi everybody, welcome to the lecture on Chapter 6, which is entitled Reproduction at the Cellular Level. And where I like to start this chapter is by showing you this picture. This is a picture of a cell under a microscope, and it's a very specific type of cell. This is a human zygote. It is the single cell that you get when a sperm cell fertilizes an egg cell. This single cell is the earliest stage of human development, and we all started out as one of these. Every single person begins as just a single cell called a zygote. And then fast forward to today, the average adult human body contains 30 trillion cells. That's the number 30 with 12 zeros after it. So obviously a lot has changed since the time when you were a zygote and today when you are a 30 trillion cell walking, talking, full-fledged person. So what happened here? How did you go from being the single cell to the form that you are today? Well, the answer is that this one cell, the zygote, made a copy of itself. One cell became two, and then each of those cells made copies of themselves, and then 2 became 4, then 4 became 8, 8 became 16, and so on and so on. And this is an actual video of this process happening to a human zygote under the microscope. It's been sped up quite a bit. The process doesn't happen as quickly as you see it happening here. Generally, it takes several days for an embryo to reach the stage where it's composed of 16 cells. And then as time goes on, those cells begin to differentiate, meaning that certain cells take on specific roles. Some become part of the skin, some become part of the bones, some become part of the heart. And over time, the embryo transforms into a fetus. And this image shows a representation of what a human looks like at various stages of early embryonic development, up to about 60 days of gestation. And all of this, of course, is driven by cell reproduction, cells making copies of themselves over and over and over again. But if this alone doesn't convince you of how important the process of cellular reproduction is, even when a human is fully grown and developed, even when you're an adult, cell reproduction doesn't stop. Your cells are constantly replenishing themselves throughout the course of your life. Old cells die, and they have to be replaced with new cells. And of course, if you're ever injured, if you scrape your knee, if you tear a muscle, if you lose some blood, all of those injuries have to be repaired through the creation of new replacement cells. So cell reproduction is important throughout our lives, not just during our early development. So in this chapter, what we are tackling is we're going to talk about how that process works. We'll look at how cells reproduce, what the life cycle of a cell looks like, how this is different in prokaryotic versus eukaryotic cells. Remember those guys from back in chapter 3? And then finally, we'll look at what happens when cell reproduction goes wrong. So, to start our look at how cells reproduce, I want you to take a moment and think, what do you imagine is the most important part of a cell that needs to be copied when creating a duplicate? There's a strong argument to be made that the most important part of the cell to copy is the DNA. Before a cell can divide, it has to make a duplicate of its DNA, because this allows both of the daughter cells, that's what they're called, daughter cells, to have a full set of genetic information that's passed down to them, which is important because the DNA contains the instructions for everything that the cell does throughout its life. Now, for our purposes in this class, we're not going to get into the detail on how this duplication process works. However, we do still need to do a quick review of what we have learned so far about DNA so that we can now build on that. So hopefully you remember that DNA is one of the main types of nucleic acids, which are one of the 4 different types of macromolecules. The other main type of nucleic acid is RNA, which we'll talk more about later. But DNA carries the information that encodes what traits an organism should have. As a macromolecule, it's made up of smaller building blocks or monomers, and those are called nucleotides. And every monomer has 3 internal components. It has a sugar molecule, which is attached to a phosphate group, and is also attached to a nitrogenous base. So this is what one DNA nucleotide looks like. We also back in Chapter 2, Part 2, looked at the process by which these nucleotide building blocks come together to create nucleic acids. So here you can see nucleotides linking together and forming a bond with each other. And when this happens over and over and over again, you get a big chain, and that's what we call the nucleic acid. Many nucleotides together is called the nucleic acid. And the length of these chains it's truly extraordinary how long they get. The largest human chromosome is made of strands of DNA containing about 250 million nucleotides, and the smallest human chromosome has strands containing 48 million nucleotides. And I say strands in the plural form because the reality of the situation is that every strand of DNA is not alone. It pairs up with a second strand, and the two strands become attached to each other through links that form between their nitrogenous bases. And those links are represented here by these little yellow lightning bolts that I've put on the screen. So we've got pairs of strands. They're made of nucleotides. They're very long, and each one of those very long double-stranded pieces of DNA is called a chromosome. A chromosome is a single, very long piece of double-stranded DNA. The image that you see on the screen here is of a chromosome from a bacterial cell as seen under a powerful microscope called an electron microscope. And it's a big mess, so you can't really tell this, but this is all one very long entangled double-stranded piece of DNA. There's just one very long tangled piece here. Now, if you remember from Chapter 3, when we talked about the physical structure of cells, you may recall that bacteria belong to a category of cells known as prokaryotic cells. Humans, on the other hand, are made of eukaryotic cells. And this distinction is important for this chapter because prokaryotic and eukaryotic cells differ in the way that they tackle cell reproduction. So to refresh your memory, I want you to think back to Chapter 3. And in this checkpoint, tell me two differences between the structure of prokaryotic cells and eukaryotic cells. Okay, so now that we've put our minds to remembering the differences between prokaryotic and eukaryotic cells, let's turn our attention back to chromosomes. Because prokaryotic and eukaryotic cells have significant differences in their chromosomes. When it comes to prokaryotic cells, they have a single chromosome per cell. That chromosome is circular, meaning that the double-stranded DNA is one really big loop. And that single circular chromosome is located, and we learned this in Chapter 3, in the nucleoid region of the cell. Because remember, prokaryotic cells don't have a nucleus. They just have a region in the cytoplasm called a nucleoid where the DNA is located. So there's no barrier, there's no special compartment to house the DNA in prokaryotic cells. But eukaryotic cells, on the other hand, they look a bit different. They have multiple chromosomes per cell instead of just one. And those chromosomes are linear in structure, meaning that the double-stranded DNA is not a loop, it's a line, and the ends of the line don't join together. And these chromosomes are located in the nucleus, which is a special compartment that's sectioned off from the rest of the cell by a membrane. And here on the right-hand side in the purple box, you can see an image of a eukaryotic chromosome, also as seen under an electron microscope. And you can see that the shape is also different from the bacterial chromosome that you see on the left, but the principle is the same. The eukaryotic chromosome, even though it's a different overall shape, it's made of one very long piece of DNA that has been tightly wound and shaped and coiled up into this distinct structure. We're going to take a look at this structure more closely in just a moment, but before we do, let's briefly go over how prokaryotic cells reproduce. And I say briefly because prokaryotic cells are so simple that they have a relatively straightforward and rapid process for division. The term for this process is called binary fission, and this term literally means splitting in two. Fission means to split. It's the opposite of the word fusion, which means to come together. So fission means to split, and then the bi in binary means two. So binary fission, splitting in two. The process is very simple. The original cell, which is called the parent cell, will replicate one of its chromosomes. Pardon me, it's one chromosome because it only has one. The chromosome as we know, is not in any sort of special compartment. It's just hanging around in the nucleoid region. And so after it replicates that chromosome, each copy of the DNA gets sent to one end of the cell, and the cell is expanding its cell membrane as this is going on in preparation of splitting in two. And this process is called segregation of the DNA. So we can see that we're in step 3 now. And then the membrane actually pinches off in the middle. So the result is that the two daughter cells are now separate from each other, and then the process is complete. So there's not really much more to binary fission than this, because the structure of the prokaryotic cell is so simple, which makes it very straightforward for them to duplicate themselves. And as a result, because binary fission is a simple process, it can occur very rapidly. So many bacteria are able to divide at a pace that is very fast compared to the more complex eukaryotic cells. So this video clip that you see on the screen shows a collection of bacteria undergoing binary fission, and the video is sped up a lot, but you can see how the cells proliferate very quickly because each time binary fission occurs, one cell becomes 2, and then each of those cells divides, so 2 becomes 4, and then 4 becomes 8, and so on. So this is a great thing for the survival of the bacteria, but for us as humans, it can be a bad thing. For example, many bacteria are infectious, and their ability to rapidly spread like this is something that we have to fight back against. For instance, and this is our second checkpoint, Proteus vulgaris is a bacterium that commonly causes urinary tract infections. And at human body temperature, it takes this bacteria about 30 minutes to undergo binary fission. 30 minutes for one cell to become 2. If a urinary tract infection starts with just 10 cells of Proteus vulgaris, after 4 hours, how many bacterial cells will there be in the infection? And as a hint for how to solve this, the number of bacterial cells is going to double for every 30-minute increment because, as we said on the previous slide, each of the new cells undergoes binary fission too. So that's the state of affairs for prokaryotic cells. Now for eukaryotic cells, things are more complicated because eukaryotic cells don't just have one chromosome, they have more than one chromosome, and they store their DNA in a nucleus, so it's sectioned off, and that introduces some additional steps as well. So how do eukaryotic cells tackle reproduction? Before we answer that question, we need to learn a few more things about eukaryotic chromosomes. So we said that eukaryotic cells have multiple chromosomes, and most eukaryotic cells have 2 copies of each of their chromosomes, one that comes from each parent. Most often there's a male parent and a female parent, and they each contribute half of the chromosomes to their offspring. These types of eukaryotic cells that have 2 copies of each chromosome, they are described as diploid, with the prefix di referring to 2. There are certain types of eukaryotic cells that only have a single copy of each chromosome, and those cells are not described as diploid, but rather as haploid. And we'll talk more about haploid cells in Chapter 7, but most eukaryotic cells are diploid. And while there are 2 copies of each chromosome in most eukaryotes, the number of the pairs of chromosomes varies quite widely depending upon what species you're talking about. So for example, this image shows a type of microscopic roundworm. This is a species known as C. elegans, and it has 3 pairs of chromosomes, or in other words, 6 total. So 3 came from its roundworm mom and 3 came from its roundworm dad. A fire salamander has 26 pairs of chromosomes, or 52 total. This adder's tongue fern has 600 pairs of chromosomes, or 1,200 total. 600 came from one parent and 600 came from the other. And domestic dogs have 39 pairs of chromosomes, or 78 total. So as you can see, lots of variation. But how do humans measure up? Humans have 23 pairs of chromosomes, or 46 total. 23 come from the biological mother and 23 come from the biological father. And here on the screen, what you see is something called a karyotype, which is an image of all of a given person's chromosomes laid out and organized as seen under a microscope. And this karyotype here corresponds to a human male. How can we tell? Well, the first 22 out of the 23 pairs of chromosomes are called autosomes. These are your regular run-of-the-mill chromosomes. These chromosomes are not related to the sex of an organism. The 23rd and final pair consists of what are called sex chromosomes, which are involved in determining biological sex. In humans, there are two possible sex chromosomes. There's the X chromosome and the Y chromosome. And generally, and this is not necessarily a rule because there are medical conditions where this can vary, but generally, having two X chromosomes corresponds to being biologically female, while having an X chromosome and a Y chromosome corresponds to being biologically male. And in our next chapter, we'll talk about some of the variations on this. But because this individual has an X chromosome and a Y chromosome, that would indicate that they are male. Now, each of these pairs of chromosomes contains genetic instructions for different characteristics. Let's talk about how this is the case. But first, you may have noticed that the eukaryotic chromosomes in this karyotype, they don't have the same shape as the example of a eukaryotic chromosome that we saw on a slide earlier. So namely, the chromosomes in the karyotype on the left kind of look like a tube, and the one on the right kind of looks like two tubes that are crisscrossing each other. So why is this? We'll answer that momentarily, but let's start by taking a look at the parts of a eukaryotic chromosome. So a eukaryotic chromosome has a distinct structure that contains two different segments called arms. There's a top arm and a bottom arm, which are connected together at a point called a centromere. And despite its name, the centromere is not located at the dead center, which means that one arm is smaller than the other. And the small arm is referred to as the P arm. The arms were named because P stands for petite. And the other arm, the large one, is called the Q arm. And that was named the Q arm because Q comes after P in the alphabet. So kind of a crude system, but that's how it was done. So together, the P arm and the Q arm, they're linked at the centromere, and that comprises the chromosome. And as we said a few moments ago, every pair of chromosomes contains instructions for a specific process. So to give you an example, let's take a closer look at human chromosome number 4. And by the way, in general, the chromosomes are numbered based on size. So chromosome number 1 is the largest one, and then number 2 is the second largest one, and so on. So on human chromosome number 4, there are an estimated 900 to 1,100 different genes. We don't know the exact number yet because they haven't all been studied. And a gene is a segment of a chromosome containing instructions for a specific thing, a specific characteristic, a process, something about the way the cell or organism operates. So remember, this chromosome is made up of one big, tightly wound piece of DNA. And within that DNA, there are all of these genes. And this diagram that you see here of human chromosome number 4, it has labels on it corresponding to some of the genes that are found on this chromosome that are involved in known diseases. So you can see labels for things like Ellis-Van Creveld syndrome, which we'll talk about later in our evolution chapter. We see labels for Huntington disease, narcolepsy, and then there's this gene sort of in the middle that I've just highlighted called SNCA. The SNCA gene contains instructions for making a protein called synuclein alpha. Synuclein alpha is an important protein that is involved in sending signals through the nervous system. Therefore, mutations in the SNCA gene can cause Parkinson's disease. Now, scientists have yet to find a cure for Parkinson's disease because fixing it would involve correcting the mutations in the DNA, which is largely beyond the capabilities of medicine at this point. But identifying the source of the disease by finding the specific gene or genes involved is the first step towards working on cures. And to keep things organized in the course of these studies, biologists have a specific system for naming the locus, which is the technical term for the location of a gene within the grand scheme of human DNA. So, for example, the locus of the SNCA gene is 4q22.1. And what this means is that the gene is located on chromosome number 4. That's what the first number means. On the Q arm of that chromosome, which is the larger of the two arms. That's what the middle letter means. And then at position 22.1, which they get by assigning numbers to different sites on the arm, and they count from the centromere outward when assigning those positions. So let's pause here for a checkpoint on this naming system. In this image on the right-hand side of the screen, you can see an illustration of human chromosome 7 with the P and the Q arms labeled, and also a number of positions of genes that are involved in different diseases. And one of those diseases is cystic fibrosis. Cystic fibrosis is a genetic condition where mucus builds up in the respiratory system, which causes breathing difficulty and frequent infections. Using the system that we just learned on the previous slide, what is the locus of the CF, the cystic fibrosis gene, that when mutated causes the disease cystic fibrosis? Okay, so getting back to the big picture, we now know the structure of a chromosome. We also know that diploid eukaryotic organisms such as humans have 2 copies of each chromosome. These matched pairs of chromosomes are called homologous chromosomes. The prefix homo means the same. So homologous chromosomes are the same chromosome. It's just that one came from the maternal side, the mom, one came from the dad. So every one of us has 2 copies of chromosome number 4. And each copy contains the same genes in the same locations. The chromosome you got from your mother contains an SNCA gene, and the chromosome that you got from your father also contains an SNCA gene. However, the content of the genes can differ between the homologous chromosomes. So your mom chromosome and your dad chromosome both have an SNCA gene, but the instructions in that gene for making the synuclein alpha protein may differ. For example, let's say that the chromosome number 4 that came from your mother contains the correct instructions for making an effective protein, but the chromosome number 4 you got from your father contains mutated instructions that result in a defective protein. So now your body is partly making the right proteins, but partly not. And this can put you at risk for Parkinson's disease. Now, getting back to that question we posed earlier, why does the chromosome on the left not look the same as the chromosome on the right? The reason is because the chromosome on the left, all the chromosomes in that karyotype, they were extracted from a cell that had not yet copied or duplicated its DNA. So chromosomes look like this before the cell has copied its DNA, but once they copy their DNA, they look like what you see on the right. So the P arm will be copied, and now there are 2 P arms. And the Q arm will also be copied, and now there are 2 Q arms, and all 4 arms remain attached at the same centromere. So it's a little bit confusing because the things you see on the left are called chromosomes, and the things you see on the right are also still called chromosomes. However, on the right, after duplication, they now consist of what are called 2 sister chromatids. A sister chromatid is one set of P arms and Q arms together. And remember, the reason that cells do this, the reason they duplicate their DNA, is because they're getting ready to reproduce, and there needs to be enough DNA to pass down to each of the daughter cells. Now, of course, as you can see, both copies of the DNA are currently stuck together in the same unit, and that's a problem. The cell is going to have to solve that and sort it out so that it can give one copy to each of the daughter cells, and we're going to see in just a moment how the cell solves this problem and separates the DNA copies. But first, just to drive home this point about DNA duplication, on the left, this is what a human karyotype looks like if you were to be looking at a cell that hasn't copied its DNA, and on the right, that's what a karyotype looks like if you're looking at a cell that has already copied its DNA. So, DNA duplication precedes cell reproduction. It comes before the cell reproduces. But when does this duplication happen in the grand scheme of things? Well, the grand scheme of a cell's reproductive process is called the cell cycle. The cell cycle represents the stages that a cell goes through from the time that it is quote-unquote born until the time that it reproduces. And as you can see in this illustration, there are several different stages that the cell goes through on its journey to reproduction. So let's briefly look at what happens in each one of those stages. The first stage of a cell's life is called G1 phase. So right after a cell is produced from its parent cell, it's considered to be in G1. During G1, the cell grows and accumulates resources. It hasn't fully committed to the division process, but it's getting ready for that. The second stage is called S phase, and it's during S phase that the cell duplicates its DNA. So this is the stage when the chromosomes go from being a structure with 2 arms to a structure with 4 arms. The DNA doubles. And that's why S phase is called S phase, because what S stands for here is synthesis. The word synthesis means to make. So during S phase, the cell synthesizes, or in other words, makes more DNA. After S phase comes G2 phase. So at this point, the cell has pretty well committed to reproduction. It has invested a ton of resources into duplicating its DNA, and now during G2, the cell will replenish its resources and duplicate all of its organelles because it also wants to pass a copy of those down to its daughter cell as well. The daughter cells don't just need DNA, they also need structures like the mitochondria and plasma reticulum, etc. Now these first 3 stages, G1, S, and G2, together, the whole thing is called interphase. And together, they represent the main portion of the cell's life. As you can see from this illustration, the cell spends a majority of its life in interphase, which is just making preparations for division. But once interphase is over, then the cell transitions into actual division processes. The division process takes place in 2 parts, the first of which is called mitosis. During mitosis, the cell is just focused on getting its DNA organized. The cell divides copies of its DNA into 2 identical nuclei, which is the plural of the word nucleus. And investing in this process of getting the DNA organized really is necessary because at the end of G2 phase, what the cell has on its hands is 1 nucleus that contains 2 sets of DNA. And mitosis is the name given to the process of the cell taking its double set of DNA and separating it out into 2 single sets, so that each single set can then be passed down to one of the 2 daughter cells. And things are further complicated by the fact that during interphase, remember interphase is G1, S, and G2 put together, the DNA is not stored in chromosome form. So instead, it's stored as these loose strands in this big, messy spaghetti bowl where it's all swirling around together with no organization. And these loose, disorganized strands are called chromatin. So here we have a simple illustration of a cell. And inside of the cell, there is a nucleus. And inside of the nucleus, you can see the DNA in the form of chromatin, which is the red and blue strands. But the DNA needs to assume the form of chromosomes if it's going to get organized, and that's one of the first things that happens as the cell moves into mitosis. The first step of mitosis is called prophase. During prophase, the loose DNA strands wind up into the chromosome form, and also the membrane that surrounds the nucleus breaks down, which effectively dumps the chromosomes out into the cytoplasm of the cell, because the cell is going to use all of the internal space that it has available to it to get these chromosomes organized. Now, as we walk through this example, we are just going to show one pair of chromosomes for the sake of simplicity, but remember that the number of chromosomes is often much, much higher than just 2 chromosomes. So in this checkpoint, tell me, if this were a human cell, instead of 2 chromosomes, there would be how many chromosomes shown? Okay, getting back to prophase, because there's still more that happens during this first stage of mitosis. So also during prophase, there are these special structures in the cell called centrioles, which I'm showing here as little green tubes, that construct things called spindle fibers across the length of the cell. And the spindle fibers are shown in orange, and these spindle fibers are going to be responsible for separating out the 2 DNA duplicates. So once those spindle fibers are constructed, the chromosomes will become attached to the spindle fibers, anchored at their centromere. And of course, this illustration is just a very highly idealized version of these events. And what they look like under the microscope is a lot more muddled. So this cell here is in the middle of prophase, and you can sort of tell that it's getting its chromosomes wound up. And then towards the end, you can probably see where the nuclear envelope breaks down and the chromosomes start to spread out a little bit more. You can't see the spindle fibers being formed here because they're too small and delicate for the microscope to pick up, but you are going to see that the role that the spindle fibers play in a couple of steps from now when we return to this same cell. So the second step of mitosis is called metaphase. And during metaphase, the chromosomes line up at the equator of the cell, while remaining attached to the spindle fibers at their centromere. The equator of the cell is exactly what it sounds like. It's the middle line. So just like the Earth has an equator that's halfway between the North and South poles, the cell has an equator too. And here is what metaphase looks like under the microscope. So you can see how the chromosomes are migrating to the middle line of the cell. The third step of mitosis is called anaphase. And during anaphase, the centromere will split, which leads the sister chromatids to be pulled apart to opposite ends of the cell by the spindle fibers. So during metaphase, the chromosomes were sitting there at the equator. They're under tension because they're being yanked on by the spindle fibers in either direction. And then as soon as the centromere breaks down, the 2 identical halves of the chromosomes are immediately pulled to opposite ends of the cell. And this is the stage where you can really see the role of the spindle fibers clearly under the microscope. So here's what anaphase looks like in a living cell. So you can see how half of the sister chromatids are being drawn in one direction and the other half are being drawn in the other direction. And this leads to the fourth and final step of mitosis, which is called telophase. During telophase, things are winding down. There are now 2 identical sets of chromosomes at either end of the cell. These will form the basis of the 2 nuclei that are going to be passed down. So a new nuclear membrane will form around each cluster, and then the chromosomes will begin to decondense and unwind back down into the loose strands known as chromatin. The spindle fibers are also disassembled because they've played their role and they're finished. And the end result of all of this is that there are now 2 identical nuclei within this one cell. And as with the other steps, here's what this looks like under the microscope. So as you can see, the chromosomes are in clusters, and you can see how they are even starting to lose that chromosome shape as the DNA is unwinding. And then you can even see the cell is actually beginning to split. So in this part of the video, we're getting a sneak peek of the very last stage of the cell cycle, which is called cytokinesis. Cytokinesis is where the one cell finally splits into 2. So each daughter cell gets one of the freshly made nuclei, as well as one of the sets of organelles that were copied back in G2. And then you end up with 2 cells that are, if everything has gone right, identical copies of each other. And now the cell reproduction process is complete. Each of these 2 daughter cells are now entering their own G1 phase and gearing up to begin another cycle. So we've been talking for a while now. It is definitely time for us to take a break with another few checkpoints to test your knowledge. So in these next two checkpoints, you're going to see images of cells captured in different stages of mitosis, and I'd like you to tell me what stage they're in. So checkpoint 5, what stage of mitosis is happening in this cell? And checkpoint 6, what stage of mitosis is happening in this cell? And then finally, checkpoint 7, what would be the consequences if the cell didn't complete S phase of the cell cycle properly? And this question is going to lead us into our next topic, which is what happens when things go wrong in the cell cycle? What happens when errors are made? Well, mistakes in cell reproduction are a big deal. They can have serious consequences for the health of the daughter cell, and not just the cell, but the overall organism that the cell belongs to. And because of this, cells take quality control very seriously. They have a built-in series of checkpoints throughout the cell cycle, which are not to be confused with the checkpoints that I ask you guys during lectures. And the purpose of these checkpoints is for the cell to check for any mistakes so that it can halt reproduction if necessary. And as we know, this is what the cell cycle looks like. And there are three main checkpoints where the cell checks for mistakes. One happens at the end of G1 phase, one happens at the end of G2 phase, and one happens right in the middle of mitosis. During the G1 checkpoint, the cell will look for several things. It's been growing and developing throughout the G1 phase, and here at this checkpoint, the cell will ask, am I now large enough to commit to the reproduction process? Because once the cell moves into S phase, it's going to start expending a bunch of resources on duplicating its DNA. Also, it will ask, have I accumulated enough of the right resources to move into S phase in the first place? And then finally, is the DNA damaged in any way? Has it incurred any mutations? Because if there are harmful mutations in the DNA, the cell doesn't want to make things worse by copying those mutations in S phase and then passing them down to the daughter cells. So if the cell passes the G1 checkpoint, it proceeds into S phase and then into G2 phase, and then at the end of the G2 phase comes another checkpoint, the G2 checkpoint, where the cell will again ask the questions, am I large enough to continue into mitosis? Have I accumulated enough of the right resources to commit to going through mitosis? And most importantly of all, was the DNA properly duplicated during S phase? Do I actually have two full and complete copies of my DNA here? And this is very important because each daughter cell needs a full set. Then if all looks good in G2, the cell moves into mitosis. And when the cell gets to the end of metaphase, which remember, metaphase is the step where all of the chromosomes are lined up at the equator of the cell. They're sitting there, they're waiting to be pulled apart. At that point, the cell will ask, are all of the sister chromatids correctly attached to the spindle fibers? Because if they aren't properly attached, then the sister chromatids will not end up getting separated and pulled to opposite ends of the cell. Instead, you can end up with both sister chromatids going in one direction or neither going in either direction. And of course, this does not set the cell up to be able to create two identical nuclei. One side will get too much DNA and the other side will get too little. So the cell has all of this quality control built in to its processes to make sure that errors don't occur during reproduction. But what happens when the quality control fails? What happens when the checkpoints miss something? What happens when damaged cells start to slip through the cracks and reproduce more damaged cells? The result of out-of-control cell division is cancer. You've all heard of cancer before. You may even know someone who's gone through cancer. But what you may not know is that cancer is a disease caused by uncontrolled cell reproduction. There are many different types of cancers. There's ones that form tumors. There's ones that don't form tumors. There are cancers of virtually every system of the body. But what unifies them all is that they are all caused by uncontrolled cell reproduction, mistakes made in the cell's quality control system, which allows damaged cells to rapidly divide. The image that you see here on the screen is a biopsy of a female cervix that is affected by cancer. Biopsy is a medical procedure that people may go through if they are suspected of having cancer, where a bit of tissue is removed and observed under the microscope to try to confirm it. So this biopsy comes from a cervix. The cervix is the lower part of the uterus, and cervical cancer is the reason why women are recommended to get pap smears every few years. They're meant to check for evidence of abnormal cell division. And then if evidence of abnormal cells show up in a pap smear, then the doctor will move to the next step and take a biopsy of the cervix and examine it under the microscope to get a more thorough look. So in this case, the cervical tissue has been dyed and colorized so that the nucleus of each cell is purple, and then the cell itself is pink. And you can probably see how on the right-hand side of the image of the biopsy, there's a very high concentration of nuclei. This means that there are cells here that are tightly packed together. They're dividing rapidly and developing into a tumor. So there's very clear evidence of cancer in this image. So how does a cell's quality control process fail? How is it that damaged cells can be allowed to slip through the cracks? Well, the checkpoints in the cell cycle are enforced by special proteins called tumor suppressor proteins. And these proteins themselves are made using instructions that are written into the cell's DNA. For example, one very well-studied example is a tumor suppressor protein called P53. The gene, the instructions for making P53 are located on human chromosome 17 on the P arm, which remember is the small arm, and at position 13.1. So here on the right, you see a diagram of human chromosome 17, and the P53 gene is highlighted. And what this P53 protein does is it checks for DNA damage during the G1 checkpoint, and if it detects DNA damage, it will halt the cycle. It will recruit and bring in other proteins to fix the damage. But if the damage can't be fixed, then it will initiate a process called apoptosis, which is basically a self-destruct sequence. The cell will self-destruct because that is the preferred option instead of continuing with reproduction and developing into cancer. So this is how P53 keeps things in check. But what if P53 stops doing its job? What if a mutation strikes in the part of the DNA that gives instructions for making P53? Now, with a mutant P53 not doing its job anymore, those damaged cells can pass the G1 checkpoint and continue on through reproduction, even though they shouldn't. And that's where the cancer risk arises, when mutations occur specifically in the parts of your DNA that are responsible for quality control. And what causes these mutations in the first place? Well, some cancers are caused by mutations that are inherited and passed down from your parents, but that is very rare. More often, the mutations are brought about by exposure to carcinogens, which are environmental agents that lead to cancer-causing mutations. And carcinogens can come in three forms. One is radiation. So, for example, UV, ultraviolet rays that are emitted by the sun, these cause mutations that lead to skin cancer. The second category is chemical carcinogens. So, for example, cigarettes contain 69 different chemicals that can cause cancerous mutations. They can not only lead to lung cancer, but also to mouth and throat cancer. And then thirdly, there are infectious agents that cause cancer. So, for example, human papillomavirus, or HPV, is a virus that goes into your cells and it changes your DNA and causes mutations. And according to the National Institute of Health, 99.7% of cases of cervical cancer are caused by an infection with human papillomavirus, or HPV. We're going to talk more about the genetics of mutation in Chapter 9. But at this point, I have another checkpoint for you related to carcinogens, specifically UV radiation as a carcinogen. So melanin is a compound found in the skin that protects against UV radiation. When skin cells are exposed to UV radiation, they begin releasing melanin. Melanin has a dark pigmentation, and this is why tanning occurs. Skin becomes darker as a protective mechanism against the UV radiation. And this phenomenon occurs in people of all racial ethnic backgrounds, but some people start with a higher baseline level of melanin that is constantly produced in their skin. And as a result, people with naturally darker skin tones are more protected against UV damage. And so we can see this borne out in the statistics where, because of their higher level of melanin, people with darker skin tones are at a significantly lower risk of skin cancer. So this table shows you the risk of melanoma, which is the most dangerous and deadly form of skin cancer. And it shows you the risk for 3 different racial ethnic groups. Black people have a 1 in 1,000 probability of acquiring melanoma over their lifetime. People who are Hispanic have a 1 in 167 probability. And white people, for this group, 1 in every 38 will acquire melanoma during their lifetime. So as you can see, there are striking differences here. However, even though Black people are at significantly lower risk for melanoma, a 2021 study found that those who do have melanoma are 25% more likely to die from their diagnosis as compared to white people who have melanoma. Why do you think this might be the case? Why might white people have a much higher rate of getting melanoma, but also a much higher rate of surviving melanoma? Now, to close out this chapter, I want to leave you with some cancer statistics for you to consider, because cancer is one of the leading causes of death in the United States. So it is something that you will probably encounter in your life at some point, whether it's through a friend or a family member who's affected by it, or even you. It is second only to cardiovascular diseases, which is heart disease. So it's second only to heart disease in terms of how many deaths it causes in the US. And in the US, the most common types of cancer are lung cancer, followed by colon cancer, followed by breast cancer, and then pancreatic cancer. Despite it being a leading cause of death, cancer death rates have fallen in the US by 27% since the year 1990. However, most of that decrease is simply because of lower rates of smoking. Tobacco use has been decreasing steadily over the past 50 years, and that has translated into huge decreases in lung cancer rates, especially among men. Another major driver of the decrease in cancer death rates is the HPV vaccination, which protects both women and men from the most dangerous strains of HPV that can cause cervical, penile, and rectal cancers. So a major takeaway from these statistics is that we haven't gotten that much better at treating cancer in recent years. As a society, we've just gotten better at preventing cancer by avoiding carcinogens. However, there have been modest increases in the 5-year survival rates for almost all types of cancer. 5-year survival rate is the rate of people who are still alive 5 years after a cancer diagnosis. So for instance, in 1999, the 5-year survival rate for lung cancer was 15%, and in 2009, it was 18.7%, so slightly better odds over 20 years. At the same time, though, the rates of some cancers are increasing in the US. And one example is the one we just talked about, melanoma skin cancer, which increased 65% between 1992 and 2019. So in some cases, it appears we're actually getting worse at avoiding certain carcinogens, like the UV radiation that causes melanoma. Of course, cancer is such a huge topic. There's entire classes dedicated to it, and there's much more that we could talk about that's beyond the scope of what we have time for here. But I do want to give you the opportunity to check out a few more graphs and statistics on cancer and to make some of your own observations. So in the next few slides, I'm going to present you with a few graphs, and I'd like you to pause the video and take them in. And for our final checkpoint, I'm asking you to come up with 2 interesting or concerning takeaways about what you see in those graphs. So the first graph here shows rates of new cancer diagnoses by age group in the US. And the number on the left-hand side is per 100,000 people. So basically what this graph is saying is out of every 100,000 30 to 34-year-olds in the US in 2020, how many were diagnosed with cancer? Out of every 100,000 35 to 40-year-olds in the US, how many were diagnosed with cancer? That's what this graph is showing. This graph shows how much the rates of cancer diagnoses have changed over time in different age groups between 1975 and 2020. So the lines are color-coded by age group, and if the line is going down, it means that that age group is being diagnosed with less cancer, and if the line is going up, it means that age group is being diagnosed with more cancer. These figures show the rates of new cancer diagnoses broken down by sex. So male is on the left and female is on the right, and then also by ethnicity. So you can see the racial ethnic groups listed on the left-hand side of the graph. And again, these are the numbers per 100,000 people belonging to that group. This graph shows cancer death rates in the US by type of cancer since the year 1930, and it's also broken down by male and female. So there's a distinct line for example, male lung cancer cases, and then there's a separate line for female lung cancer cases. And then finally, this map shows cancer death rates attributed to tobacco use, meaning out of the total number of cancer cases in each country, what percentage of those cancer cases are caused by tobacco. And as you can see from the legend at the bottom, darker color means higher percentage. So feel free to go back and pause and look more closely and find those 2 takeaways that strike you as interesting or concerning and share your reflections on those takeaways. And then once you're finished with this, you are finished with chapter 6.